Welcome back to our first TPD seminar of the year. We're very excited to have two fantastic speakers today. They'll both be sharing a talk. So our first speaker is Professor William Kaelin. He's the Sidney-Farber Professor of Medicine at Harvard Medical School and Dana-Farber Cancer Institute, a senior physician scientist at Brigham and Women's Hospital and Howard Hughes Medical Institute investigator. Among his many accolades, Dr. Kaelin received the 2019 Nobel Prize in Physiology or Medicine. He is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the National Academy of Medicine, the American Society of Clinical Investigation, and the American College of Physicians. Dr. Kaelin's research seeks to understand how mechanistically mutations affecting tumor suppressor genes cause cancer. His long-term goal is to lay the foundation for new anti-cancer therapies based on the biochemical functions of such proteins. And this was a shortened version. And our second speaker is Matt Boudreau. So Matt completed his PhD at the University of Illinois under the direction of Professor Paul Hergenrother, where he worked on a variety of new anti-cancer approaches. He is currently an NCI KOO postdoctoral fellow in Professor Kaelin's lab. His current work utilizes positive selection screening to find novel natural products that degrade crucial transcription factors found in cancer. So I will hand over to both of you, and we're excited to hear your talk.
Great, thank you very much, and thank you for that very nice introduction. So you can see my screen okay? And can you see my cursor?
Yes.
Okay, great. So my sole function today is to sort of be the brief warm-up act for Matt Boudreau. But just to give his work a little bit of context, what I thought I would share with you is that I joined the board of directors of Lilly Pharmaceuticals about 12 years ago. So I get to monitor progress or lack of progress in various therapeutic areas. And what's very clear across therapeutic areas is that therapeutic areas that are rich in targets that are genetically validated and for which we have some biological understanding of their functions are doing pretty well. So for example, in autoimmune diseases, there's a real renaissance due to targets like JAK2, IL-23, IL-17, TNF-alpha, et cetera, et cetera. That's in stark contrast to, for example, psychiatric diseases, which frankly are kind of stuck in the 1950s and 60s because we don't really have very well understood targets that are genetically validated and where we understand their biological functions. Now, a number of people have tried to quantify how important it is to have genetic validation or genetic evidence in support of your target. So here are two studies. Now, these studies focused on targets for which there was human genetic evidence, that is to say, from GWAS studies or other naturally occurring mutations in the human population. Think about things such as PCSK9 mutations leading to decreased cholesterol. But in both of these studies, they concluded that when you have a target that's genetically validated, it at least doubles your chance of being successful when it comes to developing new drugs. Now, genetic validation can actually take multiple forms, and just to make sure everyone's on the same page, by genetic validation, I mean there's some evidence that alterations in the gene lead to some alteration in a disease-relevant phenotype. So again, the two earlier studies I just showed you, they focused primarily on human variants and mutations. Of course, in cancer, we often rely on recurrent somatic mutations in various tumors. But genetic validation can also come in the form of well done, I'll say properly controlled somatic knockout or knockdown studies, such as now using CRISPR-Cas9 or recently with RNA interference. And of course, there are also in some cases where we rely on mouse knockout studies to provide genetic validation. Now, many of us worship at the altar of DepMap, and we certainly do. So for example, here are data from DepMap looking at depletion or enrichment for guides against the HIF2 transcription factor across various tumor types. And I think you can see that a subset of kidney tumors are highly dependent on HIF2. Frankly, we had come to this conclusion using horse and buggy technology many years ago, but it was nice to see it in DepMap. And you may be aware that just recently a HIF2 inhibitor has now been approved for the treatment of kidney cancers, at least in the context of VHL disease, and it's anticipated this first-in-class HIF2 inhibitor will also soon be approved for sporadic kidney cancer. So if we look at recent cancer drug approvals, you can see that in almost all cases, the targets conform to what I was talking about a moment ago, that we had targets that were genetically validated, and we had some biological understanding of why those mutations would actually play a causal role in cancer. And so the outliers are things like, for example, the proteasome inhibitors that are now used in multiple myeloma, where there was never really any genetic validation in support of the proteasome as a target of myeloma, and really only on a post hoc basis that people come up with hand-waving explanations for why proteasome inhibitors might be useful for the treatment of multiple myeloma. So in the field of drug discovery, people are certainly moving in the direction of genetically validated targets and away from sort of empiric targets. Now, you might be surprised to see CTLA-4, for example, here, the immunotherapy target in this circle, but I will remind you that for the immune checkpoint targets, we actually did have genetic evidence, at least in the mouse, that these were important rheostats on the immune system. So earlier studies had shown that knockout of CTLA-4 in the mouse causes autoimmune phenomenon, and that was sort of, again, the evidence that this was a rheostat on the immune system, and led a number of people, including Jim Allison and Gordon Freeman, to suspect that if we could inhibit some of these so-called immune checkpoints, they would be useful in cancer. Now, some of us are old enough to remember when the New York Times proclaimed that Judah Folkman was going to cure cancer with chemicals such as endostatin and angiostatin, but in the end, these went nowhere. And I would point out these had no genetic validation linking them to angiogenesis and cancer, and how they actually operated as angiogenesis inhibitors was pretty unclear. But fortunately, later came along the VEGF inhibitors, and at least in the case of VEGF, there was clear genetic validation that VEGF controlled angiogenesis in vivo, and that VEGF was a growth factor and survival factor for endothelial cells. So that brings us to the problem that, as this audience well knows, there are many examples of high-value targets in cancer that are genetically validated, that are pretty well understood biologically, but where we've deemed them to be undruggable. We all have our favorite list. Here's one of my list. I'm hopeful that maybe RAS will soon be leaving this list, but this still leaves some high-value targets that are undruggable. Now, how can we drug the undruggable? Well, in some cases, you can go downstream of the undruggable target, exploiting epistatic relationships. In some cases, you can develop allosteric inhibitors. There's a renewed interest in allosteric inhibitors. In fact, that HIF2 inhibitor I alluded to a moment ago is actually an allosteric inhibitor. I've had a long-standing interest in exploiting synthetic lethal interactions to drug the undruggable. And of course, as this audience knows, there's now a renaissance in terms of interest in targeting undruggable proteins with degraders. So I think, as a number of you on the call appreciate, a number of years ago, our lab and Ben Ebert's lab, working in parallel, showed that the IMiD-like drugs act as so-called molecular glues that recruit the cereblon ubiquitin ligase to target two undruggable transcription factors, IKZF1 and IKZF3, for degradation, and we linked that to the anti-myeloma activity of the IMiDs. Shortly thereafter, people like Jay Bradner and Craig Cruz showed that you could actually modify these molecular glues to now recruit new targets. But in fairness, this paradigm kind of is reminiscent of this older idea that was championed by Ray Deshays and Craig Cruz years ago, that you might be able to develop bifunctional small molecules that would recruit E3 ligases to degrade specific targets. And I think this audience is pretty aware that there's some jargon here, that these smaller, more drug-like molecules like the IMiDs are sometimes referred to as the molecular glues, whereas these molecules on the left are referred to as PROTACs. But if you step back, of course, there are lots of other ways you could imagine a small molecule drug directly or indirectly affecting the stability of a protein. And I say that because, of course, protein stability is highly regulated. So this audience probably knows there are about 500 ubiquitin ligases and about 100 DUBs, and they regulate the stability of various proteins. But their ability to engage those proteins or recognize those proteins is often influenced by things such as protein post-translational modifications, protein folding, interacting proteins, et cetera, et cetera. So we wanted to develop an assay that was sort of agnostic with respect to how a small molecule was degrading a protein. And one of the mantras in my laboratory is that up assays outperform down assays. This is something I was taught many years ago by colleagues in the pharmaceutical industry who do a lot of high throughput screens. And the reasons for this are fairly straightforward. First of all, up assays usually have better signal to noise characteristics. It's easier to see a positive in a sea of negatives than a negative in a sea of positives. If you think about it, this is what makes astronomy possible. But more importantly, screeners, and these, I'm now referring to people who do chemical screens or genetic screens, also prefer up assays, because if you do a down assay, there's this constant headwind of uninteresting, trivial positives, false positives, and that's because there are lots of uninteresting ways to make a complex system work worse than better. And so when in doubt, we try to do up assays. Now, I think in previous seminars in this series, we've talked about an up assay that was developed by Sagar Kaduri when he was in my laboratory. And the idea here is to have a protein of interest fused via a little spacer to a modified cytidine kinase that can accept a non-natural nucleoside and convert it into a toxin. There's a V5 tag, and then off of the same transcript, there's also a GFP reporter. And so the idea was you could add a chemical or genetic perturbant, add BBDU, and now look for green survivors. And so in a proof of concept experiment, Sagar did 384 well plate assay. So here the cells have this suicide protein fused to the IMiD target IKZF1. And in each well, we're adding a different chemical. And I think you can see there are at least 3 positives. So these 2 positives over here are pomalidomide, a professional IMiD. This well actually contains a chemical that actually interferes with BBDU uptake. So it's simply an assay positive. Sagar went on to screen small libraries of annotated chemicals. So I apologize for this busy slide, but the top plate has the IMiD target IKZF1. The bottom plate is the counter screen with the unfused DCK. There are some controls in the outer columns that you can ignore for now. But now, otherwise, each row has 2 chemicals at 10 different concentrations. So for example, here's an assay positive that's scored in both plates, so we don't care about that. Here's a true positive called Spaltin-1, which promoted the survival of these cells in a dose-dependent fashion, and in validation assays that Sagar subsequently published, this turns out to be a true positive. So again, we like this up assay for looking for degraders. It actually lends itself to doing pooled CRISPR screens, and Matt Ozer, a former postdoc on my lab, has used this technology to do pooled CRISPR screens. But we also thought, being an up assay, this assay might lend itself to screening not only annotated chemical compound libraries, but also screening complex mixtures of natural products. And I think that's where Matt is now going to take over.
Little shuffle. Awesome. Thanks, Bill. Check find the mouse. All right, awesome. Yeah, so Bill just introduced this up assay for protein degradation that Sagar Kaduri, former postdoc in our lab, had developed. So I'll just quickly go through this just in the sense of time. So this utilizes this modified deoxycytidine kinase developed by the Medin lab. And by fusing that to our protein of interest, and upon treatment with BBDU, you'll see no GFP signal. And then in this case, you would see no GFP survivors. But you can imagine, either in a genetic experiment or a chemical screen, if that condition led to the downregulation or degradation of your protein of interest fused to DCK, and then upon treatment with BBDU, you would then be able to find these green survivors. And again, we see this in 384. Where you see where positives are the green here. So, and again, Sagar, in the initial report in 2021, they had shown that you could use this to find to annotate different imid derivatives and their efficacy towards IKZF1, and also, as Bill pointed out, was able to find spoutin-1 as a novel IKZF1 downregulator degrader. And so I'll give a shout out to the Dana-Farber TBD seminar series. So Sagar and Matt Ozer. Sagar had done a lot of the chemical screening initially with the IKZF1 system, and Matt Ozer had done some lineage-specific small cell lung cancer studies using CRISPR screening. And so they gave a really beautiful talk. I actually saw this before I was a postdoc, and I've loved this seminar series for many years. And so I'll point people to that seminar. It's a really great overview of the DCK system and how we think about it. So as Bill said, we really have been thinking about the big targets in cancer and how we can utilize this up assay system to start thinking about how to address them. And so today, I'll specifically be talking about my efforts to utilize the DCK system to establish beta-catenin in that system. Is there a way? Okay, sorry. Just use the perfect keyboard. Okay, great. Sorry, just getting used to this setup. Awesome. So again, this is the DCK V5 IRS GFP system, and then we clone in beta-catenin fused to DCK, or in the same plasmid but lacking DCK, we have just beta-catenin overexpress. So the system looks something like this, and I'll point out a couple of things that we've innovated on since the last time we've talked publicly about the DCK system. So this is all in a 293T background. These cells are not sensitive to BBDU. You can see that by the black curve here. We now utilize an extra gene knockout in these cells, so thymidine kinase 1 knockout or TK1 knockout. We find that when you knock out TK1 and you express DCK, those cells are extra sensitive to BBDU. And so this is a new knockout that we utilize for our system to really increase that signal to noise and really bring these curves down to where now we can think about screening with lower amounts of BBDU, where previously with Sagar's work, we had screened in the 100 to 500 micromolar BBDU range. So that's a new feature in the system. You can see that in our cells, TK1 is knocked out. In this case, we see that beta-catenin fused to DCK cells are sensitive to BBDU, and that isn't something going on just by sheerly overexpressing beta-catenin, which you can see with the red curve. And importantly, when we express these transcription factors fused to DCK, we want to make sure they're still functional. And so AXIN2 is a bona fide target gene for beta-catenin. And you can see that the DCK fused to beta-catenin and just beta-catenin alone overexpress. You see that we see similar AXIN2 levels, indicating that protein is most likely folded correctly and functional. So with this, we're a little bit more optimization than this, but for the interest of time, I won't talk about that. But I'll jump right into our high throughput screen assay and how that typically looks. So we utilize the ICCB facility at Harvard Medical School, where we plate cells with the combi. We allow them to attach overnight, and then we actually use acoustic dosing. This is a new instrument that we utilize for liquid handling over at ICCB, which is this echo compound transfer, where we dispense 100 nanoliters of compound, and then we wait for 24-hour incubation, and then we dose in BBDU, and then we wait 4 days. And we're looking for assay plates that look like this. Z prime is a statistical measure between your positive and negative controls. And you can see that our assay is quite clear, where the positives are very green, that the negatives are dark. And for the Z prime aficionados out there, greater than 0.3 for a cell-based assay is really great. For us, our Z primes are always well above that 0.3 metric at 0.5. And so, again, just to reiterate kind of what Bill had said previously, we do a head-to-head screen. So we have beta-catenin DCK cells, and then we have DCK alone. And we're looking for the wells that selectively score in beta-catenin cells and not in DCK. So here, the red arrows indicate shared positives, so those would be uninteresting. And then the white arrow, again, would be something that would be interesting, because it seems that well affects beta-catenin cells specifically. So with that assay in hand, we really were thinking about what kinds of things should we screen for to go after these classically undruggable targets. And I always gain a lot of excitement and inspiration from the way natural products have interesting, and previously you wouldn't expect those mechanisms, you wouldn't pull those mechanisms out a priori, right? So I always like to point out Taxol. I think if you told an incoming graduate student to think of how a molecule can kill a cancer cell, you likely wouldn't come up with a stabilizer for microtubules. And nature, however, has spent many years establishing that mechanism as a potent way to kill cells. So Taxol is a great example, but I think there are many, especially those that come directly from microbial extracts, things like FK506 or rapamycin that glue, that bind FKBP12 and then glue to something like calcineurin in the case of FK506. For rapamycin, of course, that's mTOR. But there's also a variety of other examples from the fungal natural product space where you can now glue 1433 to a variety of proteins of interest. And those molecules have been really great case studies for how molecules can lead to proximity. And of course, in the plant hormone space, and especially in the targeted protein degradation application, things like auxin and brasolinolide, where they now, it's very similar to things like pomalidomide and other IMiDs. They bind an E3 ligase that leads to the degradation of a target. So we know that nature is really good at making these very intriguing natural products, and those natural products often have very interesting and unexpected activities. And we know that natural products are a fruitful chemical space, as there are many drugs, not only for cancer, but for other indications that either are directly natural products or derivatives of natural products going forward. So natural products are a very fruitful ground for finding new mechanisms and attacking challenging therapeutic targets. But unfortunately, we don't typically use them at high throughput screens anymore. So this is just a quick way of looking at this, just looking at published high throughput screens. And you can see that most high throughput screens denoted in gray utilize annotated small molecules. Typically, these are synthetic libraries or known bioactives. And very few of those published screens actually utilize natural product extracts directly to use in the high throughput screen. And so there's a lot of reasons for that, and I think we have a little bit of time today to really discuss some of these. I know this can be a little wordy, but I think we could give a whole seminar probably on this one slide. But I'd like to just point out some key features that our natural product library that we have access to really seeks to address. So the first problem is having access to a lot of material and a lot of diverse natural product material. So you can imagine that in the case of Taxol is a great example. So that was isolated from a bark from a plant, and the yields were extremely low. So if you wanted to utilize Taxol, the major bottleneck there was actually getting enough material and yield from the actual parent natural product material. So access to that, having it be reproducible is a huge problem. So of course, then we have the problem that now we have access to material, but often that's a complex mixture, and there's a lot of just general cytotoxicity that's found in these complex mixtures. And so you can start to think while complex mixture is quite challenging for an up assay, that toxicity would simply just not show up, and then we could actually find those green survivors for an up assay. So we'll come back to that throughout this talk. With that complex mixture and just general promiscuity, typically when you screen natural products in a high throughput screen, they're extremely high hit rates, and that can be really challenging going forward. So if you imagine doing a really large screen, and even a 1% hit rate can be really challenging based on how big that hit rate can really gain. And that really can be derived from a lot of reasons. And one of those is that these natural product extracts often have a lot of pan-assay interference compounds, or what we traditionally refer to as pains. And getting those pains out and identifying those pains really early in your hit validation is really important, because if you run the screen, you get a lot of pain molecules, you really don't want to waste your time on those. And so that's something to keep in mind for these natural product extracts. Poor reproducibility is endemic in natural product screens, and that really can be thought of in two ways. One, getting the same molecule out of the natural product material can be really challenging, and it's important that you have checkpoints there to make sure that you're actually accessing the same molecule. But also things like viscosity. So you can imagine a natural product extract, sometimes the viscosity is quite a problem. So if we're doing something like acoustic dosing, where that viscosity really becomes important, we really need to have solid reproducible results, and this is something that happens a lot with natural product extracts. Of course, we don't know what natural products are in these extracts, so it can often be challenging, especially given the complexity of natural products, to ultimately deconvolute what the active pharmaceutical or small molecule that's actually in the well. And even if you do spend a lot of time to deconvolute them, often you'll just rediscover known natural products. And so you need to have a way of quickly assessing whether a hit is something that's already known. And so it's typically referred to as a term called dereplication. And something that I won't spend a lot of time on, but really could be its own seminar, is ethical and legal sourcing of primary extracts. So a lot of effort and bureaucracy is now established for gaining access to natural products and their extracts from different countries, and making sure that the host country intellectual property and considerations are maintained. So there's now a lot of treaties and bureaucracy that supports ethical and legal sourcing of these natural products. So the NCI has a natural product division that's very interested in trying to have a screenable library that overcomes a lot of these. And this has been a huge endeavor taken on by Barry O'Keefe at the NCI, where they now have what's called the Natural Product Discovery Pre-Fractionation Library. So we take these really, really complicated primary extracts and we pre-fractionate them. And we'll talk about that and how that was designed to kind of help some of these challenges that we just discussed. And so now this pre-fractionated library can be utilized and screened. And so a huge shout out not only to the ICCBL Harvard Medical School facility, but also the Ludwig Cancer Research Initiative. So they provided a lot of institutional support to actually reformat the entire natural product library into a screenable format for ECHO at the ICCB. And so there's a huge investment from these two, and I really want to give them credit. So let's talk a little bit about the fractionation of these natural products. So historically, the NCI has a large extract repository, mainly coming from plants, marine sources, and microorganisms. And what Barry's team established was a way of making a generalizable fractionation strategy where you actually load that extract onto cotton balls to get that to really nicely have a nice beginning to the solid phase extraction. So you load that extract onto a cotton ball, you then stuff that into a traditional C8 SPE column or solid phase extraction. And that column serves a couple different purposes. One is just we're going to end up separating based on polarity, but it also, the resin that is utilized in that SPE column removes a lot of tannins and other pain molecules. And we'll talk a little bit more about why that's important. And then we're using an eluent that's a mixture of methanol and water. And so from at current numbers, this is a little out of date now, but utilizing the natural product repository of the NCI, they've conducted now a fully automated SPE fractionation of those, and we have currently access to over 320,000 of them. This is now actually closer to 500,000 that's available from the NCI. And so just because this will be important as we go through, every parent extract, or F0, has a corresponding 7 fractions. So when you screen this library, you get information not only from the primary extract, or F0, but you also get those 7 fractions. And we'll talk that through with kind of our efforts with beta-catenin. Some more things about this. So now every fraction has reduced chemical mixture complexity. So there's still mixtures as a fraction. On average, 10 to 15 compounds per fraction. Of course, that varies depending on where the extract is coming from, whether it's a plant or a marine extract. Again, this C8 SPE removes a lot of those tannin and phenolic-based pan-assay interference compounds. And often that fractionation does, for our purposes, does 2 things. The traditional sense is that fractionated extracts typically show enhanced biological activity. So those things like minor metabolites are concentrated in fractions, so you're more likely to find them scoring. For us, it also, since we have an up assay, we need cells to survive, it also removes just some of those general cytotoxic compounds that you can find, especially many of those are phenolic panes. And so that fractionation really also helps our up assay perform better. So this is also housed at the NCI. They have a huge compound repository now. And from this, where now we can very quickly deconvolute a lot of these extracts, and we'll talk through their strategy for supporting that. And because it's all highly automated, and a lot of the sample preps, columns, HPLCs are all fully automated, and so it's highly reproducible access to materials. So when you say you want to access a certain fraction, they'll then pull from their library, and you still have maintained activity from that. So I'll just point people to a review that Barry and his team published. They talk about a lot of these challenges in making the prefractionated library and just general challenges of developing assays that'll work with things like complex natural products. So they're kind of the most important part, right, is that can we get to a compound at the end? And I think that can be really challenging. I think we can all appreciate that. And so there's a full ACS Chem Bio paper that describes their deconvolution process. So I'll point you there, but we'll do a little quick run through today. So traditionally, you screen the prefractionated library. Again, that's 1 parent extract per 7 fractions. You conduct your high throughput screen. You have your list of hits. I can hide it. Yep, sure. Hold on 1 second. Perfect. Okay. So you run your high throughput screen, you have your list of hits, and then you go back to the NCI. They then take those list of fractions and you conduct a semi-preparative HPLC method that converts that 1 fraction into 22 new subfractions. Typically, these are much less complex than their parent fraction. So this is just an example of what one of those traces can look like. You can see, for example, subfraction 12 and 13. You can imagine that being actually quite pure, whereas something like a subfraction 8 might be a little bit challenging. But we have all this information, including IR, at this stage, and we can dereplicate from known IR stretches at that point. So you then have your 22 subfractions from your active fraction. You then rescreen those, and you get your subfractions that score. You can then go back to the NCI, where now that we know which subfraction is the active component, we can do a variety of other HPLC methods to isolate those compounds specifically. You can then re-get those. We typically get those in about 2 to 5 milligrams of scale, so we have plenty to actually find activity. And then ultimately, you can arrive at natural products that are the active component. So these are just 2 published examples that I'll just show today. One in Cell Chem Bio, one in ACS Chem Bio. But these are the kind of structures that they pulled out of the lab so far, and there's numerous other unpublished examples, but we'll leave these for today. So we have access now at ICCB to screen this natural product library, and we, as Bill mentioned, we now have this up assay that can be utilized to find compounds that lead to the downregulation of your target of interest. And so now we really feel like we have a platform that we can find natural products that affect oncogenic beta-catenin, and we can just marry these 2 together to really try to find these really intriguing compounds. So I'll describe a lot of work in one slide here, but this is the entire high throughput screen that we conducted. So we screened 326,000 fractions from the NCI library. Utilizing these hit metrics is z-score greater than 3 in beta-catenin and less than 1 in DCK, because again, we want ours to be specific to beta-catenin cells. The primary hit rate from that screen is 0.46. Again, this screen includes things like F0s, those parent extracts, and their fractions. So we actually filter out hits that only scored in the F0. So deconvoluting from F0 is quite challenging. And so we just go ahead and filter those out. So really from the fractionation, we have 736. And then upon further cherry picking and reconfirmation, we arrive at 353 confirmed hits in total from the high throughput screen, which puts our hit rate at about 0.1, which is quite respectable, not only just for a high throughput screen, but also for a natural product high throughput screen. And so these 353, I'd like to just point out a couple of vignettes about them. The first of which is where they're coming from. So the current incarnation of the library is about 3/4 terrestrial plants. The next majority is marine, and there's a small subset that are marine plants and fungal microbials. From our screening, our 353 confirmed hits, they broadly recapitulate the origins of the library, and so we don't see any biasing, which I think is interesting and also good. And so that's kind of what they look like from an origin perspective. I really also like to see just where they've come from. So each star represents 1 or more active fraction. So our 353 hits come from a total of 70 different world regions, which just again speaks to the diversity that we have in the NCI Natural Product Library. The top 3 countries here, the United States, we have 49 hits. 35 of those come from Madagascar, and 30 of those come from Tanzania. And with Catherine's really nice. invitation for us to speak today, I thought it would be important to highlight our hits that are from New Zealand. And so we have 18 marine hits actually coming from both islands in New Zealand. I'm just selecting here. Most of them are marine sponges. And so we actually have some really exciting hits that are coming from New Zealand. I thought it'd be good to point those out. So I spent a lot of time introducing this pre-fractionation, and I just want to show what you can get in terms of information from the library. So most natural product libraries, you simply just screen the parent extract. So that would be F0. So this extract, 90865, if we had screened this in traditional mode, just F0, that would not have been a hit. So remember, our hit needs to be 3 for the red bar and less than 1 for the blue bar. However, when you actually screen fractions from this parent extract, we do see 2 fractions, 5 and 6, that score really nicely and very selectively between DCK and beta-catenin. And we have many examples where this is true, where the fraction is scoring, but not the F0. And what's really reassuring, especially from a chemical perspective, most of these fractionations are not optimized for the active component. And so it's really reassuring to see adjacent fractions scoring, especially at this early stage. You can even see, maybe not statistically relevant, but at least the trending. You can even see that maybe it bleeds into F7. And so this, again, just highlights, this is just one example. We have obviously many examples from the library where the fractionation is very important. But just in total, for all hit fractions, 84% of them, the F0 showed no activity. So again, speaking to the power of why pre-fractionation can lead to enhanced biological activity, whether that's from concentration of your active component or whether that's removing toxic compounds out. So now you can allow the fraction to score in a positive selection screen. So another aspect is we do get some information generally about the polarity of our hits. So we're in a cell-based screen. So unsurprisingly, things like fraction 1 and 2 and 3, which predominate with very polar compounds, likely lack cell permeability to score in this assay. But we do have a significant bias towards 5 and 6. Of course, increasing lipophilicity increases cellular promiscuity as well as permeability. And so this is something to keep in mind. Of course, many organic natural products will probably lie in this fraction 5 and 6 space. But so we do see a significant bias towards 5, 6, and 7. So now we've run the high throughput screen, and now we're at the point where now we're conducting subfractionation. So we just finished the entire screen actually right before Christmas in early December. We're still getting a lot of the subfractions to begin this next part. But you can imagine that before you do the 300,000 extracts, we'd want to kind of test run this. And so that's exactly what we did. So after the first 51,000 extracts, we then arrived at 29 hits, and we utilized those 29 hits to test this workflow. So those 29 hits for 22 subfractions per hit, you arrive at 638 subfractions. And from those, we rescreened those comparing DCK to beta-catenin cells, just like before. And we arrived at 22 subfractions that showed selective activity in beta-catenin cells and did very little to DCK alone cells. And those are highlighted here. You can see that there are some that are adjacently scoring, which is always nice to see. And I'd like to just drill down into these for a second. So now we have the extract ID here, and you can start to see where these lineages are coming from. So L15009 is a terrestrial plant extract where the fraction 5 and fraction 6 scored. And interestingly, of the subfraction, which utilizes the same HPLC method, the same subfraction scored, likely indicating the same compound that scored in fraction 5 also scores in fraction 6, and then robustly scores in fraction 7 of the subfraction. I think it's really nice to see here in the data. And again, we do see many others that have, these aren't optimized for the active component at this stage. So we do see adjacent fractions scoring quite regularly at this stage. So from these 22 subfractions, we actually have enough to do a Western. So this is my stressed out slide. So this would be considered the current world supply of this natural product subfraction at this stage. The liquid line is here. But there's enough to get a Western blot. And we actually were able to utilize that for all subfractions hits and arrive at 4 subfraction sets that actually do something to beta-catenin at the Western blot stage. So when we talk about those 4 of our initial test set, so 3 of these are terrestrial plants, and one is a marine sample from Palau. And many of these did exactly what we thought they would do. So I'm just showing here the L15009 series. Remember, this is where the fraction 5 and 6 are scoring, and then the shared subfraction 7 is the active subfraction. And you can see on Western blot that beta-catenin is going down. This is at 24 and 48 hours. And importantly, not only is beta-catenin going down, both exogenous and endogenous, but the target gene, axin 2 for beta-catenin, is going down. So those two vectors are in the same direction. Just a control for just general proteome. I like to utilize ERK1 and 2, and we don't see a change there. It seems to be specifically beta-catenin going down, axin 2 target gene going down, and does so at 24 and also at 48 hours. Another one that surprised us on the blot is this one. So this is from the pencil cactus from Tanzania. And this extract, this is the one that spanned over all of those fractions. But you can see that instead of down-regulating beta-catenin, which is kind of how we developed the DCK assay to find molecules that would down-regulate proteins of interest, this molecule actually appears as a subfraction, I should say, appears to lead to increase in beta-catenin expression, both exogenous and endogenous. But even though there's more active beta-catenin, there's less target gene. So axin 2 is down. So now these vectors are in opposite directions. But if you look at the fold change, it's actually quite intriguing. So this is something we weren't expecting, but I think this is something that we are really interested in, is it seems to imply that maybe beta-catenin is aggregated. We have some preliminary evidence with these subfractions that indeed that we are making punctated beta-catenin in the cytosol, therefore not active in the nucleus. So those two and a couple of the other ones were looking very interesting. So now we go to the next phase of this, which is utilizing the NCI for characterization support. And so we take all of these subfractions from these four families we were interested in, and now we convert them into what we call P numbers, or deeper purification. And so we have P numbers that correspond to that lineage, and I've just highlighted that here on the left. And we compare their activity in beta-catenin cells versus DCK alone cells, so the same screening assay that we normally do, and now we're able to do a dose response. Now, these are pure to semi-pure. So some of these P numbers are 100% pure. We know what the structures are. I just won't have time today to talk about some of those. But I'll just highlight today the L15009 lineage, so P27990. This one, we know the chemical structure for, we're pretty close to it, but unfortunately the supply has been quite challenging. So I won't talk too much about that molecule, but that molecule does lead to the downregulation of beta-catenin in a couple of different contexts. L16943 is this one coming from the stabilizer or aggregator lineage. And so this molecule 27980 is the one that's really performing well. You can see there's a couple other ones that look good here, but in other secondary assays, the true active component is this molecule. At this stage, at least when these slides were made, this molecule is about 50 to 80% pure, which is a pretty significant range. But you can see in a Western blot, it does lead to that stabilization that we saw at the subfraction stage, does downregulate axin 2. And it does lead to aggregation or increase in beta-catenin at a quite potent concentration, as sub-micromolar. Again, our current hypothesis, and we have some evidence for this, is it appears to be leading to punctated beta-catenin in the cytosol. And we currently think that's why it's able to score in the screen. And obviously, we're pretty interested in this molecule and trying to elucidate its structure as we currently speak. So we have some hits on the structure, but it's not quite ready for prime time quite yet. So, and that's exactly where we are. So we are now getting subfractions for the rest of the hits. So I just showed you subfractions from 28 or 29. And so now we have way more subfractions to go through, way more molecules to find, and hopefully more single molecules that do something to beta-catenin. So I think that's where I'll leave it for today, and really excited to answer questions as well. Obviously, a huge thanks to Bill, his constant support through this entire process. Big shout out also to a research technician that's been working with me, helping me move a lot of plates. Sophie has been really great. And of course, the entire ICCBL screening team, as well as the natural products branch, Barry O'Keefe's team, and there's a lot of support there. And so with that, I'll take any questions.